Welcome to another session of the Women's Health Seminar Series. Today, it is my distinct pleasure to welcome Dr. Kieran O'Donnell. I am going to tell you a little bit about him in just a second, but before I do, I want to give our land acknowledgement that we recognize that we live, work, play, and participate on a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of the work that we do, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, which is covered by Toronto Purchase number 13 with a Mississaugas of the credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members also live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. And in case you don't know who we are it's free to join, so please join us. We have over 750 members across 32 countries. We have a number of events, including this Women's Health Seminar Series. We have some online conferences, some how-tos. We have a YouTube channel with over 600 subscribers. We do research in multiple diverse fields, and part of our work here is to empower those who study women's health research, find information, and empower those to bring sort of a community of us together. We do a lot of outreach. We have some trainee travel awards. I believe we've given out over $30,000 in travel awards over the years. So please do apply for those when you see them. And obviously, we can do this without support from our sponsors. So thank you to them. And please do become a member. And with that, I'll stop sharing so that Dr. O'Donnell can share his screen and I'll introduce him. Dr. O'Donnell is an assistant professor jointly appointed within the Yale Child Study Center and the Department of Obstetrics, Gynecology, and Reproductive Sciences at the Yale School of Medicine. Dr. O'Donnell leads the Health-Omics and Perinatal Epidemiology. Why can't I say words? Oh my gosh. Epidemiology Research Group, which seeks to better understand individual differences in the fetal origins of mental health. In 2019, Dr. O'Donnell was elected to be a fellow to the Canadian Institute of Advanced Research in the Child and Brain Development Program, and he was also awarded a NARSAD Young Investigator Award and named a Jacobs Foundation Young Scholar. And you'll see why very soon, because he does amazing work. In 2022, Dr. O'Donnell was named a Scialog. What's that? That's not how you pronounce it. Fellow, well, how do you pronounce it?
Scialog.
Scialog, a molecular basis of cognition program from the Research Corporation for the Advancement of Science. He also serves on the Advisory Council of Postpartum Support International, a leading perinatal mental health training and advocacy organization. I'm so pleased that Dr. O'Donnell agreed to do this. He does some amazing work, and as you heard, has an impressive CV and record and body of work. I always like to give out fun facts so we don't, so we know the other side of our researchers. And Dr. O'Donnell was great and gave me a bunch of fun facts. And there's so many that I have to read at least a few of them. He is father to a super cute 15-year-old boy. And although he might be biased, I have seen pictures. And so I would also verify, independent verification, that he is very super cute. He has a slight obsession with CrossFit, which apparently is basically a playground for big kids. I'm not sure that that's true. I've heard it's really hard. And he comes from Ireland. And if you've seen the Banshees of Inisherin, that's basically where he is from. And hopefully it's not so colorful as that film. In terms of the characters were colorful and dark. Uh,
Sure.
and I wouldn't say that Dr. O'Donnell is dark, let's just say. That I know of.
Well, Dr. Galea, thank you so much for that invitation. And I will say, in terms of whether or not I am dark, there's an interesting duality. Another fun fact that I didn't tell you about my name. So Kieran in Sanskrit means ray of light, a ray of sunlight. But Kieran in Irish, Gaelic, which is where I'm from, means little dark one. So perhaps there's something to that. So good afternoon, everyone. As Dr. Galea mentioned, I'm based here in the Yale School of Medicine, but I wanted to start just in solidarity with anyone who is not currently at the American College of Neuropsychopharmacology meeting. My social media feed is just blown up with people at ACMP. And so here is a picture of the beautiful Arizonan desert for those of you who, like me, are not able to make that meeting. And for those of you who are joining us, thank you very much for taking time out of your sightseeing trip, I mean your scientific meeting, to come and join us today. So I've been working in the area of perinatal mental health since around 2005, first at Imperial College in London and then at McGill University, and now currently based here in the Yale School of Medicine, where I hold joint primary appointments in the Child Study Center, which is essentially our Department of Child and Adolescent Psychology, Psychology and Neuroscience, and then also in the Department of Obstetrics, Gynecology and Reproductive Sciences. And the reason for that joint appointment is because my research program is really focused on trying to understand the predictors of maternal perinatal health and more broadly maternal perinatal health, and also how perinatal health can impact the health and well-being of the next generation. And we're particularly interested in trying to leverage information from different biological systems to improve the prediction of health states. Now, in terms of being working in this area of perinatal mental health, it is actually quite an exciting time. So this year in the United States, we had the first national strategy to improve maternal mental health care. We've had announcements from the Biden-Harris administration about increased funding for maternal perinatal mental health. And then more recently, we've had the launch of the HRSA maternal mental health line, which is available in multiple languages, 24/7 in English and Spanish, with I think something like 60 other language represented through the availability of interpreter services. And then in Canada, we will very soon have the release of CANMAC guidelines for perinatal mental health. So there was a great presentation from Patricia Tomasi from the Canadian Perinatal Mental Health Collaborative outlining the work of this committee, that I imagine the publication will be forthcoming. And of course, as Dr. Galea mentioned, there's numerous initiatives in Canada and internationally that are seeking to improve the health and well-being of women and families. And why is this? Well, partly because of the profound human suffering and the neglect and underrepresentation of women in science and in research. And also, and this is perhaps from a colder perspective, because of the very large economic costs associated with untreated women's health issues, particularly in this cost analysis from the United States, untreated perinatal mood and anxiety disorders, where there's an estimated cost of approximately 18 billion US dollars per year. Similar cost analyses have now been performed in the United Kingdom and in Canada, really highlighting the very large economic impact of untreated perinatal mood and anxiety disorders. And, working in this space, there are numerous studies. It's very difficult to keep up with the literature now with the pace of publication on perinatal mood and anxiety disorders and its impact on the next generation. But every now and again, there's a paper that will stop you in your tracks and you'll think, gosh, that is something that is surprising. And this is one of those papers that I highlight a lot because I think it is quite profound. It's work from Rebecca Pearson in the ALSPAC cohort. So this is the Avon longitudinal study of parents and children. And so what's unique about this cohort, and this is a cohort that I've worked with myself since my PhD, is that they've tracked health and well-being for around 15,000 pregnant individuals across pregnancy, now across early childhood, adolescence, and now these adult offspring are having pregnancies of their own. So this is looking at the prevalence of prenatal depression across two generations, so mothers and their daughters. And so when you look at daughters of women who didn't endorse high symptoms of depression during pregnancy, approximately 16% of those daughters went on to report high levels of depression in their own pregnancy. So what about the daughters of women who did endorse high symptoms of depression in pregnancy. Well, over half of those endorsed high symptoms of prenatal depression in their own pregnancy. And there's two striking points about this study. One is that overall, the study reported an increase in the prevalence of prenatal depression across generations. So that overall prevalence was increased, but also highlighted the marked increase in prevalence as a function of exposure to maternal depression. Now, one point that I want to point out is that this is based on a very small sample size. So even though it's a remarkable cohort, 15,000 pregnancies, obviously they are adding to their numbers as each daughter gives birth and goes through pregnancy themselves. But still, this analysis was based on around 180 individuals. But as I mentioned, they're recruiting continuously into this cohort. And so we've recently reanalyzed their data with approximately double the numbers, and the findings hold up. We see that around 45% of daughters of mothers who experienced high levels of depression in pregnancy go on to report high levels of depression in their own pregnancy with similar findings for depression. Now, it's at this point in my talk that normally I would segue into the impacts on the child in terms of the impact of untreated perinatal mood and anxiety disorders on child outcomes. And for today's talk, in the interest of time, we're going to focus a little bit more on the biological embedding, on measures of biological aging. So this is a key point that I want to bring up upfront at this point in my talk, is that preventing or treating maternal perinatal anxiety and depression is an important and critical clinical endpoint. Irrespective of any impact on the next generation and the health and well-being of the family, preventing and treating maternal perinatal anxiety and depression is the key clinical endpoint. And of course, if we can develop interventions that can improve maternal mental health, which of course there are interventions now that exist to do so, then improving the health and well-being of the next generation is a potential important secondary outcome. And I will say that we do require increased experimental evidence to verify this. So I think the use of randomized controlled trial is really going to be critical to answer this important question. So one of the findings from our research and the research of most groups working in this area is that there are marked individual differences in the impact of maternal perinatal mood and anxiety on child outcomes. And similarly, there are marked individual differences in trajectories of maternal perinatal mood and anxiety symptoms. And the question is, how can we get at this issue of individual differences? Are there any biological systems that can be informative for predicting individual differences in symptom trajectories in both maternal and child health outcomes? And so one system that we spend a lot of time interrogating ourselves in my group is that of the DNA methylome. So DNA methylation as a mechanism that can alter the function of the genome. And so this is a review that I was fortunate to be part of with my Canadian Institute for Advanced Research colleagues, where we evaluate the evidence for DNA methylation and other epigenetic modifications as candidates for the biological embedding of experience. So what I mean by that is trying to understand how our experiences get under the skin. What are the biological processes that are sensitive to the environments that we inhabit that can influence our health and well-being across the lifespan? And DNA methylation is perhaps the most well-studied modification in this class of epigenetic modifications, partly because it's very stable and can be measured in archived blood samples and other tissues. We know that it can be functionally relevant, so it can alter the amount of gene expression from a given gene. And we also know that there are now standardized methods of analysis. That means that you can pool data across multiple cohorts to investigate or develop epigenetic biomarkers, which we'll talk about, that are not as sensitive to the side effects that you might see with, for example, neuroimaging data. Now, I mentioned that DNA methylation is a functional modification. And so we can think of the cells that we collect for our biological analyses as having a unique epigenetic signature. So we know that DNA methylation varies across cells. And so when we collect a blood sample or a cheek swab sample, we're collecting thousands of cells that each can have different levels of DNA methylation at given sites. So really what we're looking at is a mosaic that we need to interrogate and estimate DNA methylation across multiple cells and derive an average then of DNA methylation at given cells. And so the typical metaphor that's used to explain DNA methylation is that it's a little bit like a light switch. So you have the light being turned on with gene being expressed, or the light being turned off with the gene being repressed. And we know that from work by Jenny Tung and others, Elizabeth Binder, and many individuals working in this field, that it's a much more complicated association between DNA methylation and gene regulation. So context is critically important. Where DNA methylation is occurring can really determine whether or not it increases gene expression, whether it decreases gene expression, or whether it has no impact at all on gene expression. Um, and using DNA methylation, so keeping that in mind in terms of its functional relevance, there is the capacity to develop epigenetic biomarkers. And in our field of epigenetic epidemiology, I think the most successful application has been in the development of epigenetic biomarkers of aging. So work by Steve Horvath, Morgan Levine, and Dan Belsky and others have shown that there are predictable and consistent changes in DNA methylation that accrue across the lifespan. And you can use that information on variation in DNA methylation at age-associated sites to derive measures of epigenetic age. And the first suite of epigenetic biomarkers of aging were simply trained to predict chronological age. So what are those age-associated changes in DNA methylation? Can we use that information then to derive a measure of epigenetic age? And what has emerged from these analyses is that individuals that show accelerated epigenetic age, so that's higher epigenetic age relative to chronological age, have higher risk of all-cause mortality in older age, have decreased cognitive function and increased cognitive decline, and are at risk for other adverse age-associated health conditions such as cardiovascular disease. And so the geroscience definition of aging is a progressive decline across organ systems. And when we think about these epigenetic biomarkers, essentially they are proposed as biomarkers of the molecular damage associated with aging. So the acquisition of molecular damage associated with aging. Now, up until recently, most of the epigenetic biomarkers of aging that were developed using samples from older cohorts or cohorts that are overrepresented in midlife and older age. And so together with colleagues, we were involved in developing an epigenetic biomarker of aging that could be applied to pediatric samples. And so this is work that was led by Lisa McEwen in Mike Cobor's lab, that brought together a large collection of data sets that spanned the pediatric age range from 0 to 20 years of age. And so just under 2,000 individuals. And critically, some of these data sets had longitudinal data. So essentially using the same tools and techniques that have been applied to develop epigenetic biomarkers of aging in older cohorts, the same tools and techniques were applied, lasting regression, to identify those those sites where we see age-associated changes in DNA methylation. And then we can use that information to derive measures of epigenetic age for individuals. And so we can benchmark that against existing epigenetic biomarkers of aging and test whether or not this pediatric specific biomarker of aging better predicts chronological age than the older established epigenetic biomarkers of aging. And so this is what you get when we look at one of our longitudinal data sets. So this was a data set where we had longitudinal data at from around 5 to 7 years of age, and then again from around 7 to around 12 years of age. And so when each individual is represented by a line, and you're looking at that change in epigenetic age as a function of chronological age. Now, what we should expect to see are positive slopes. All of these children are getting older over time, so all of those slopes should be positive. Now, I think what you can appreciate from this plot is what we would describe as it's a mess, because all of those slopes are all over the place, and that is the application of the existing epigenetic biomarker of aging, the Horvath clock, that was trained in cohorts that really underrepresented the pediatric age range. And so we see that there's an error of around 3.8 years in the prediction of chronological age, which is obviously a very long time in the lifespan of a child. And so this is what we get when we apply the pediatric epigenetic biomarker of aging. So this is trained on pediatric samples. This is developed using a training data set and then a test data set that's completely independent. And so when we apply this pediatric epigenetic biomarker of aging, you can see that the error dramatically decreases. So it's down to around 6 months is the error in this prediction. And you can see that objectively the slopes for all of those individuals become more positive. So we're seeing what we would expect, that the epigenetic age is increasing over time in line with the chronological age increasing over time. Now, of course, the question is, why is any of this relevant and why do you care? So if we can just ask someone what their birth date is, or ask a caregiver what their birth date is, and then if we can just record the data which you've collected, the clinical data of interest, well, then you have someone's age. So does this biomarker of aging tell you anything beyond what we can glean from someone's age? And so that is the question that we sought to test in this is now published by a couple of years ago in biological psychiatry, work by a very talented student, Megan McGill, when I was back at McGill. And this is taking DNA methylation data from two independent cohorts. One is a mixed ancestry cohort. It's from Singapore. So we have individuals of Indian, Malay, and Chinese ancestry. And then we also have a cohort from the Netherlands, which was who were predominantly identified as white or European. We have DNA methylation data across 3 time points in the context of the Singaporean cohort from 3, 9, and 48 months. And then we have data at 6 and 10 years of age from the BIBO cohort, the Dutch cohort. And essentially the setup for the model is that we're testing the impact of prenatal anxiety on epigenetic age acceleration across time using mixed models. We've also done this using generalized estimating equations and get very similar results. And we're adjusting for gestational age at birth, maternal education, and then cellular heterogeneity, because as I mentioned to you before, epigenetic modifications can show cell type specificity. So controlling or considering the heterogeneity in your biosample of interest is an important consideration. And so what we found in this paper was that prenatal anxiety was associated with accelerated epigenetic aging. We saw this across both cohorts, and these effects seem to strengthen over time in terms of the Singaporean cohort, the younger cohort. We had a stronger impact of anxiety over depression. But what I wanted to share with you today, because this is all published data and you can, of course, read this for yourself, is that the BIBO cohort is a longitudinal cohort that is continuing. They have collected biological samples now into adolescence. And we're particularly interested in adolescence as a potential sensitive period. There's some evidence that the impact of adversity can be readjusted or recalibrated. And this is work by Megan Gunner suggesting that adolescence is a period where there can be recalibration, particularly of the hypothalamic pituitary adrenal axis. We were interested to know whether or not there could be any recalibration of epigenetic age in adolescence. So making use of data from their 14-year time point, we've asked quite simply, just do the effects of prenatal anxiety on epigenetic aging persist over time, or is there the potential for this recalibration effect? And I should also mention that in our earlier published work, we found a marked sex difference in the impacts of prenatal anxiety on epigenetic aging. These effects were only evident in males, they were not evident in females. And so looking at the new unpublished data, we find pretty much exactly the same thing. So essentially, there's a positive, significant association between maternal prenatal anxiety and epigenetic aging in boys, that is completely absent in females. And so the red line denotes essentially the line that we would think of as being of clinical, of significance. So basically where this effect size becomes significant, and you can see that that falls quite below the established kind of clinical threshold for clinical anxiety of clinical concern, which would be approximately 40 on the Spielberger trait anxiety inventory. So we're seeing that this seems to be a linear impact of prenatal anxiety on epigenetic aging that survives adjustment for socioeconomic status and other covariates of relevance. So again, finding no evidence of a recalibration on the epigenome during adolescence. As in just a little side note, I discussed these findings with Megan Gunner, who's involved in this study. And she kind of looked at me and she said, "Well, why did you expect a calibration?" And she pointed out that in her work, where she does see a calibration, these are individuals that have been adopted into high socioeconomic status households, having experienced institutionalization early in life. So these individuals have had a dramatic shift in their social environments, and then you see this recalibration effect. Essentially, all of the children that we've been investigating in this longitudinal perspective cohort are growing in place. They're living in the homes that they've lived in all of their lives. So it's perhaps not surprising that we don't see a recalibration effect. The sex-specific mechanism is really something that we are trying to make progress on. So our initial hypothesis about potential differences in inflammation and immune cell infiltration into the oral cavity haven't borne out. So we find no differences, no sex differences in the infiltration of immune cells into the oral cavity, which was one of our pet hypotheses as to what could explain sex differences in epigenetic aging as a function of prenatal anxiety. So the hunt for a mechanism continues, but the hunt for more relevant biomarkers also continues. I think one of the limitations of the biomarker that I've just described to you is that essentially it's trained to predict chronological age. Now, when we think about aging, there are likely to be pathological, damaging changes in DNA methylation that are associated with aging, but there are also possibly adaptive changes in aging. If you think about a cell trying to survive, perhaps in adverse circumstances, then there may be changes in the molecular state of that cell that promote survival of that cell that we could think of as adaptive changes. And so the clock that I've just described to you doesn't parse adaptive from pathological changes. But when we think about childhood, there are multiple processes that are going on. We have growth, so we have organ assembly, there's the growth of the system, then there's the development, the maturation of those systems. And then in parallel, we have aging occurring, the acquisition of molecular damage. And so these clocks that we're working on that look at chronological age, that are trained to predict chronological age, they're picking up on multiple, potentially distinct biological processes and creating one summary index. And perhaps if we were able to parse these different processes, we'd be able to get a better sense of how the prenatal environment or other sources of adversity or potential sources of resilience are impacting these processes. And there has been some progress in trying to parse these effects using adult clocks that have been applied to adult samples. This is work from Vadim Gladyshev, which looks at what he refers to as causality enriched epigenetic age biomarkers, where they're trying to decouple the damaging changes in DNA methylation associated with age and then the adaptive changes in DNA methylation associated with age. Now, one key requirement to be able to do this in childhood is the availability of large data resources and the requisite measures of biology. And I think the HBCD cohort and the ECHO initiative are both initiatives that I think will be informative for trying to develop more refined epigenetic biomarkers of child growth, development, and aging. And of course, there has been the development of what we refer to as second and next generation epigenetic clocks, again, largely applied to cohorts of adults and trained and developed in cohorts in midlife to older life, and then used to predict health outcomes in midlife to older life. And so I just wanted to introduce you to some of these second and next generation clocks, and then talk about some of the work that we've been doing to test whether or not these clocks can reveal the impact of pregnancy on system health. So there are one of the first second generation clocks that was developed by Morgan Levine, a former colleague here from Yale, who's now moved to Altus Labs. And essentially, this was the motivation behind this was to create a better biomarker of mortality rather than just using age-associated changes in DNA methylation, using information that was gleaned from clinical tests, particularly blood tests, testing which of those blood parameters were associated with mortality, and then creating a phenotypic age and asking which DNA methylation sites predicted that phenotypic age. And you can see the components that went into this biological age estimator, this pheno age. And so essentially there's 513 CPGs, 513 sites that can then predict this phenotypic age that was shown to better predict all-cause mortality and a range of other adverse age-associated health outcomes. Now, and of course, we refer to this as a next generation or a second generation epigenetic clock because the first generation were trained to predict chronological age. Now, this is trained to predict, it's essentially a mortality risk estimator based on these clinical parameters. This next epigenetic clock was developed by Steve Horvath and Anki Liu. It's called Grim Age. So the workflow is essentially trying to identify which clinical parameters, again, using blood tests in the Framingham Heart Study and then validated in the Jackson Heart Study, which of these proteins from blood tests are associated with all-cause mortality, and then creating an epigenetic biomarker of these proteins that then can be summed together in one overall biomarker. And Steve Horvath describes this as the Grim Age clock, with high values being Grim Age because it is perhaps one of the best predictors of all-cause mortality of any of the epigenetic biomarkers of aging. So we have phenoage, we have phenoage, and then we move to what we refer to as next generation biomarkers of aging. So essentially, the first generation biomarkers of aging trained to predict chronological age cross-sectionally. Second generation biomarkers of aging essentially trained to predict mortality risk using proteins identified in blood and again using a cross-sectional analysis. Then Dan Belsky made use of the Dunedin cohort in New Zealand, where they looked at age-related change in multiple different phenotypes, 19 different biomarkers of aging across a 20-year period. And then they asked, which are the DNA methylation sites that can predict these individual trajectories of change across these multiple biomarkers? And they derived a biomarker of the pace of aging referred to as Dunedin pace. And so when you look at values for the second generation biomarkers of aging, these are expressed in years. When you're looking at Dunedin pace, it's seen as a ratio. So essentially, if you're scoring a Dunedin pace of 1, that means that for every chronological age, every year of chronological age, you're adding 1 biological year. And so obviously, if you're adding 1.2, you're adding 20% extra per year for your biological aging. So of course, the question then is whether or not we can use these second and next generation biomarkers of aging as integrative measures of system health. So they've associated with cardiovascular disease, one of the leading causes of maternal mortality. The question is whether or not these biomarkers may be sensitive to the state and the progression of pregnancy. And so pregnancy has been described as a natural stress test. And so we know that in individuals that experience pregnancy complications, for example, have a higher future risk of cardiovascular disease. And these effects can emerge as early as 4 years postpartum. And so the American Heart Association has described pregnancy as a potential model for studying the impacts of this physiological stress as a window into future cardiovascular disease risk. But there's an interesting review in Trends in Endocrinology and Metabolism that counters this idea that pregnancy is just quite simply a physiological stressor, and also highlighting the potential pro-rejuvenating effects of pregnancy. So we know there can be increased mobilization of stem cells in pregnancy, and we know that there can be many beneficial impacts of pregnancy on system health. So perhaps it's better to conceptualize it as a yin and yang, where you have these pro-aging physiological stressors, and countering that, we have these pro-rejuvenating, anti-aging impacts of pregnancy. So what is the evidence that pregnancy can accelerate biological aging? Well, this has been quite the year for studies on pregnancy and impacts on biological aging, really led off by fantastic work by Jesse Boganik and Vadim Gladyshev. So using multiple different systems and models, they showed that physiological stress is associated with accelerated biological aging, but there seemed to be a recovery. Once this physiological stress had passed, there appeared to be a reversal of biological age across multiple different stressors and systems. But what really caught my attention was their work on pregnancy. And so using mouse data, they showed quite nicely that there was an increase in biological age across pregnancy, and there seemed to be a reversal in the postpartum. And then using publicly available data sets, they showed really quite clear evidence for increases in biological age across pregnancy. And then one cohort of 14 individuals where they had measures of biological age in the postpartum, and they found a suggestion that there was a potential reversal using one of these second generation biomarkers of aging, the pheno-age clock. So around the same time, we were working with Claudia Buss and Elizabeth Binder with a really unique cohort from the University of California, Irvine, where we had repeated blood samples in early, mid, and late pregnancy, and critically at 3 months postpartum. So we had the longitudinal data across pregnancy in around 110 pregnant individuals, and then the 3 months postpartum data in around 68 of those individuals that came back for follow-up. Now, this cohort was from recruited in Orange County in California, pretty well resourced group, and their average age was around 28 years at recruitment into the study. Now, my hypothesis when I first reached out to Elizabeth to set up this collaboration was that we would see an impact of pregnancy on biological aging that would just accelerate biological aging. But I really didn't anticipate the marked reversal in biological age that we observed in the postpartum. And this was fantastic work led by Hung Pham in my group that really complemented and extended the work that Jesse Boganik had published earlier in Cell Metabolism, showing that yes, indeed, there is a significant impact of pregnancy on biological aging. So we see increased biological age going from early, mid to late pregnancy that exceeds the passage of time based on chronological age. But then there's a marked decrease in biological age in the postpartum. So you can see, for example, with the Grim age estimator, we're going from 4.6 for 46.5 years in late pregnancy to 43.5. Pheno-age shows perhaps the largest change, so going from 37.8 years to 31.4 years. And then the pace of aging, you can see that there's a marked decrease. Again, you're going from kind of increased or accelerated pace of aging to a decreased pace of aging from late pregnancy to 3 months. And I will say that for most of these biomarkers, we also see a significant difference between 3 months and early pregnancy, suggesting that there is overall, on average, a decrease in biological age that exists between 3 months postpartum and early pregnancy. But what I want you to appreciate from the figures on the left are the marked individual differences in this reversal effect. And increase while we see kind of a narrower clustering of biological aging across pregnancy, there is really quite dramatic differences across individuals in terms of that postpartum recovery effect. So overall, on average, we see this decrease, but there are many individuals that don't show as marked a decrease as others. Now, we were very interested in trying to determine what would be potential predictors of this postpartum recovery effect. And so in this study, what we were very fortunate to have were a number of phenotypes available to us to test for associations. But the two that we were most interested in testing for associations were maternal body mass index and also breastfeeding or nursing. And the reason for that is because metabolism and metabolic activity are perhaps some of the best predictors of biological aging, both in the preclinical literature and in the human literature. And so what we found was that high pre-pregnancy BMI was associated with a less pronounced recovery in the postpartum, such that individuals with a high pre-pregnancy BMI had higher biological age estimates in the postpartum. And in contrast, what we found was that individuals that reported exclusive breastfeeding from 0 to 3 months showed a more accelerated decrease in biological age from late pregnancy to 3 months postpartum. And so pointing to two phenotypes of particular relevance for influencing trajectories of biological age over time. Now, I think when the study came out, there was a lot of discussion about what are the whether or not this is confounded by cell type. So we know that there are marked and profound changes in cell type across pregnancy that necessitate the establishment and maintenance of pregnancy. And we see this in our own data. So these are data that we included in our paper, where you can see that there are dramatic changes in cell type composition across pregnancy. But even when we adjust for cell type differences, we still see an impact of BMI on multiple of the measures of biological age, even adjusting for cell type, also for other relevant covariates, including obstetric complications and socioeconomic status. So of course, this paper received a bit of attention. It was, I will say that the first title for the Science article was "Pregnancy Takes Years Off Your Life," and thankfully they got the hopeful message that we were focusing more on reversal, and it was edited slightly. And then around 2 weeks later, a really impressive paper came out from Kaylin Ryan, who's at Columbia, working with the Cebu cohort in the Philippines. And this was a really comprehensive study in a much larger cohort than we had available to us. And seemingly it suggested contrasting or conflicting findings with us, suggesting that the greater number of pregnancies was associated with increased epigenetic aging, which kind of countered what we were proposing, which was that there was an increase in biological age over time, but that there seemed to be this reversal in the postpartum. And I want to highlight this figure from Kaylin's paper, and just to highlight a couple of key differences between our cohorts. So Kaylin's cohort, I think the average age was around 21 years of age at the baseline, and many individuals had already experienced pregnancies at this point in life. There were individuals that had as many as 6 pregnancies in their cohort. They came from areas where there was could be relatively poor access to healthcare and health coverage, and we don't know the nutritional environment that of these pregnant individuals, and also just to highlight the marked individual differences. So you have individuals that have 3 or more pregnancies, and they have high biological age. That means that on average, as a group, they have higher biological age relative to those individuals with no pregnancies. But you can see that there's really a dramatic spread. So there's certain individuals with 3 or more pregnancies that actually have lower biological age than individuals that have no pregnancies. So I think the take-home message from both of our studies is that there's marked individual differences in the impact of pregnancy on biological age, but that there is a critical important need to consider context. And so this is a small study that embarrassingly I was alerted to after we had published our paper, that looked in a very small cohort of individuals. Each individual is represented in a line here, where they look at epigenetic age in mid-pregnancy and then 1 year postpartum in a US-based cohort. And again, they find that there's an overall decrease in epigenetic age going from mid-pregnancy to 1 year postpartum. Again, speaking to potentially there being context-specific effects of pregnancy on biological aging. Now, just a couple of points to note. In a similar vein to how I described the limitations of the application of pediatric epigenetic clocks to studying growth, development, and aging, it's important to note that most of these epigenetic biomarkers of aging, the second generation epigenetic clocks particularly, were trained to predict mortality. And so there is a question about whether or not pregnancy as a state is associated with mortality and premature death. And so it is interesting to do a review on the literature linking parity or pregnancy to mortality and to premature death. And I was quite surprised with the studies that I was able to find, which essentially showed that the effects of parity on mortality are really only observed at the extremes, at very high parity. So this is one really interesting study in a group of Amish individuals where they looked at mortality effects. So one of the kind of limitations or confounds associated with many of these studies is socioeconomic status, other co-occurring risk factors. So this was in a clearly defined population group with similar socioeconomic status. And what they found was that the impact of parity on mortality was really only observed at 14 or greater pregnancies. And then this is the study on the right, Barclay and colleagues, is from an interesting Swedish cohort where they also had adoptive parents included in their analysis of biological parents. And the take-home message there was that there's really seemingly a protective impact of parenting on mortality, but that there might be a slight uptick in mortality risk at higher parity levels, and so a decrease in relative life expectancy when you get to seven or more pregnancies. So I think that again, context is important, but also parity does not necessarily equate into decreased mortality risk. I think where these mortality risk estimators can be particularly useful is when they're applied to populations that have higher mortality risk, particularly in the perinatal period. And I think where we have more scope to use these biomarkers in the context of the perinatal spaces, particularly here in the United States, to try and use these biomarkers to test whether or not they're of value for identifying individuals that are at risk of maternal morbidity and mortality. And of course, we know that this is patterned by race, such that individuals who are Black or African American experience a disproportionate burden of maternal mortality and morbidity here in the United States. And of course, Arlene Geronimus has described this idea of weathering, so the chronic stress associated with racism and discrimination essentially wears the biological system down, leading to increased risk of age-related disorders in individuals, in younger individuals. And so I think this is going to be an area that we can use these mortality risk estimators to test whether or not they're of value for predicting health states. And so I'm collaborating currently with Dr. Alison Hipwell and Ashley Hill in the Pittsburgh Girls Study to essentially test that hypothesis. And just one last point to mention is that, moving forward, I think the next frontier moving from kind of single measures of mortality risk is really towards a more multi-system biomarker. So trying to test how pregnancy can influence biological aging across multiple systems. And of course, there's been some fabulous work done by Winnie Orchard and Helena Rutherford and others working in the UK Biobank, testing for parity effects on measures of brain age and really showing that the effects of parity on age-related changes in brain structure and function actually are somewhat opposite to the effects so that there's an opposite effect of parity to aging on these measures of brain age and aging. So I think that it's interesting to see the extension of these aging studies to the maternal brain. And of course, the fabulous work by Emily Jacobs and of course, Susanna Carmona looking at changes in grey matter density, CSF volume across pregnancy and into the postpartum. So I think really the next frontier is how we try and combine information across multiple systems to really reveal the full impact of pregnancy on biological aging across systems. And there are some promising new tools at our disposal to test this. So this is an example from Albert Higgins Chen, a colleague here at Yale, who has a nice preprint describing systems age. And so essentially they develop measures of biological age across multiple systems So in terms of the brain, lung, liver, musculoskeletal, so there's 11 different systems that they have. They claim that they can assess measures of biological age that then, of course, you can combine into a combined systems age, but it does give you that more nuanced analysis of biological age across multiple systems. And one of the really nice examples that they provide are three individuals. So what you're looking at in this spider plot or radial plot are measures of biological age across three individuals that are matched in chronological age, that are matched in systems age, that composite score. But when you look at the individual components of that systems age score, thank you, you basically see that there are marked individual differences in the specific systems that are contributing to this biological age score. So just to conclude, I think that what we've seen is that pregnancy associated effects on biological age may be reversed, but there's really lots of work to do. So what I haven't talked to you about today is how changes in steroid hormones impact changes in biological age. So we have those data available to us. I can tell you it's a little bit complicated, but I think it speaks to this idea that there are individual differences in the impact of pregnancy on specific biological systems that then means that the contribution of steroids to that process is must be done at the individual level. And of course, the question is the stability of these effects of pregnancy and biological age over time. So is this reversal effect a true reversal effect? Does it exceed preconception levels? Is it of relevance for maternal health outcomes? These are all questions that we're seeking to address, particularly in the Pittsburgh Girls studies, but also more in intervention cohorts. And I just wanted to reiterate the point that while pregnancy is an experience that's shared by all pregnant individuals, the impact of pregnancy on specific biological systems can vary across individuals. And I think having the ability to interrogate and probe different biological systems will allow us to test how pregnancy can impact the function and health of those systems. And that is likely to vary across individuals. And ultimately, to be able to do that, to be able to develop those tools, we need to have increased research for women's health research. And so these are just two examples that I think really nicely make the argument for why that's important. And so with that, I'd like to thank all the individuals that have made this work possible. Thank you for joining us today. And thank you, Dr. Kalia, for the invitation to speak at this event.
Thank you so much. I know it's always hard with these virtual events, but if some of you online can give some like little emojis, that'd be great. We have about 8 minutes for questions, but that was fantastic. A real tour de force. And actually, you've taught me so many things about epigenetics, and I took a whole bunch of pictures and like the clocks because of course I knew about the second generation, but it was really great because that helps us understand, especially when we're not in the area, why there might be some differences. Um, I am going to look at the chat for questions, but I'm going to take the liberty, it's the best, of asking the first questions and more questions if none show up. And so a question I have always been interested in is the biological clocks, and you can really pick any of them. I was quickly looking
Yeah,
at the second generation because it was great because you showed me like here are what the signals are looking at, or this is what they're looking at.
yeah.
Um, I'm just wondering how much of that is based on more male biased data.
Mm,
So I think I looked at like creatine. I always say that wrong, and my son
yeah,
always gets mad at me. But it does look like it's more. It goes down more in males with age than in females with age. So if they're
yeah.
biased, if the clocks are biased because the signals are more like of a signal in males rather than females, we might see something else in data exclusively from females and aging.
Exactly, exactly. So I think that really speaks to the point. So one of the best examples there is leptin. You get really
Yes,
marked
leptin. Yeah,
differences
yeah,
in leptin
yeah,
in these biological clocks. So
yeah.
obviously with the Grim age estimator, you can pull out the subcomponents. And so we did
Okay.
that with our pregnancy
Oh, did you? Yeah,
analyses,
nice.
just trying to test which
Yeah,
were there
which
any of the
ones?
subcomponents
Yeah,
that really showed an impact of pregnancy. We didn't find one that was
any of
like
them.
the aha moment. Oh,
Yes,
this is clearly the
right.
the reason why we see these effects. But yeah, so with leptin, you see marked sex differences in leptin, which is quite nice because you would expect to see sex differences in leptin on the basis of the data that's been published on the peptide itself. But
Yeah.
I think to your point, it's critical that we think about how they're to develop and what are the biomarkers that would
Hmm.
inform the development of female-specific
Hmm.
biomarkers of aging.
Hmm.
Um,
And
and I think, sorry, go ahead.
Kaylin has her, yeah, yeah. But what before we get to Kaylin, what about fetal sex? Did you? I think you did.
Yeah,
Uh,
yeah. So yes, so I remember many years ago, I gave Claudia Buss a very hard time because she presented work that suggested that there was like this fetal sex effect on maternal cortisol, and
Why'd
then
you give her a hard time?
anyway, I don't think the result held up. So that in my
Okay,
defense.
okay,
Um,
okay, okay.
but
Logic.
yeah, and since then I've always been really interested in thinking about
Yeah,
whether or not fetal sex can influence
yeah,
maternal
yeah,
physiology.
yeah, yeah.
And we didn't find any
Didn't
sex
find any.
difference
Because
in
yeah,
biological aging.
yeah, it's interesting, and I think if you're talking about different organs, you might there maybe, but that's
Yeah.
but anyway, we could talk for a long, long time, and I could just email you and talk to you. So I will let Kayla. Did you want to unmute yourself? Oh, there you
Yeah,
are.
Kaylin.
hi, Kieran. Thanks so much. This is an excellent talk. It's very cool to see it all in one place. My question is actually not about the maternal side for now because we can talk about that another time. I'm curious about how you're thinking about the pediatric clocks, and
Yeah.
something that sort of I've been bumping up against is how do you interpret them? Do you know, because accelerated, you mentioned this a little bit, accelerated age in the pediatric clocks, but like, do you know of people doing work to find ways to link those to things that we care about, like health or growth or things that might be something we can make them more tractable about, I guess.
Yeah.
You have a
Yeah.
boy. Have you noticed the growth charts for boys versus girls? Just thinking
Well,
about.
yeah,
No.
actually, I mean, and I think what's interesting though is
Sorry.
I might be wrong, but boys don't necessarily show the deviations, at least in the adult epigenetic clocks, until around after puberty. So you sort of see a switch there, it looks like, but I'll let
Yeah,
I'll let
Yeah.
yeah.
you.
So we actually see in that Bebo cohort, we see sex differences emerge in epigenetic age using the PED-BE, that pediatric specific clock at around 10 years of age. And so we do have puberty data available in that cohort that we can test whether or not it's actually reflecting the timing and tempo of sexual maturation. In terms of trying to link it with phenotypes, obviously, we're interested in trying to test whether or not these measures of epigenetic age are associated with child health outcomes. And so in the Bebo cohort, we found that, again, only in males, there was an association between epigenetic age and mental health symptoms in boys, but not girls. But I think trying to take it that one step further, where we actually kind of train a new kind of suite of tools is something that I do think that we as a group need to get together to do because most of the cohorts that I'm working with, I don't think are adequately powered individually to be able to develop these clocks. I think even the PED-BE initiative that was led by Mike Cobor, like that took a lot of work just to get age and DNA methylation from that many cohorts. I think trying to get other potential biomarkers of inflammation, potential neuroimaging measures, like I think that's where the HBCD cohort will be helpful in the echo cohort because they're harmonizing measures and they'll have them, ideally in the thousands. But I think it's something fun to think about how would we do this and what are the measures of growth, development, and aging that we should prioritize.
Thanks so much.
Thank you for your question. Kristen.
Oh, hello. That was a really great talk. Thank you so much. So when you're talking about DNA methylation, you're looking at global DNA methylation, right?
So we're looking at site-specific DNA methylation
Okay.
across the genome. So it's around in the more recent ways that we analyze the data, we get around almost 1 million sites. But then of those, only a few hundred go into these epigenetic biomarkers of aging.
So yeah, what I was thinking about is, have you looked at any histone marks specifically, like methylation of the histone marks? Because a lot of I think those are kind of sexually dimorphic in the regulation, right?
Yeah.
And then you also have the protective factor of, I think it's H3, K27, I don't remember, in the placenta during pregnancy that kind of specifically protects females. And so this may be more of a cell type specific question, but just
Yeah.
wondering about the contribution of those kind of in the
We,
intergenerational transfer.
Well, in theory, anything that shows changes with age, any molecular feature that shows changes in age can be used to inform the prediction of, or the development of an epigenetic biomarker of aging. And there are actually some histone clocks out there. We haven't tested any of them ourselves. One thing that we have done is there's an RNA age calculator. And so we did apply that to an RNA sequencing data set that we had, and we found that there was just a really high error rate in it. And I think it really depends on the size of the training data set that's been used. So I think in theory, that's a really good idea. And another reason why you'd need to have a very large funding envelope to be able to look at these six specific epigenetic biomarkers of aging. I think it would be a really fun thing to explore.
Cool. Thank you.
Well, thank you so much. That is pretty perfect in terms of answering questions there, Kieran, from people that, in terms of our timing. I'm sorry that is all the time that we have today, but I just join me again in thanking Dr. O'Donnell for a fantastic talk. I have so many more questions. I've got a page full of questions, but maybe I might save you by you know, taking a picture and sending it to you or something like that. Or, the next time we meet in
Next time we do it in person,
yes, I'll
that'll
bring
be
it
the
with
deal.
me and then I'll press you for all the questions. So thank you so much for a great talk and for your advocacy. That was really fantastic. Did you see me doing this at the
I
end?
did, yeah. Thanks. I
Anyway,
appreciated that. I
no, that was
did.
great. Thank you very much. And goodbye,
Great. Thank you
everyone.
so
Thank
much. Thank you all.